Okay, room. So for the most part, I'm going to let you guys start by kind of leading this discussion. If anybody has any questions that they'd like to throw out, if you would like to speak up, we can also do it chat style. If you want to throw questions into chat, that's also completely fine. I've pulled up all of the lectures via PowerPoint. Hey, you will be rewarded for attending this meeting. I almost want to stop the recording so folks who are not here don't get that kind of information. But yeah, I will try to kind of direct you towards the most important material you should be focused on, and also kind of the level of understanding that you should be hitting on it. And again, I think that's kind of the most useful thing about a review session. I am going to have to limit it to an hour. So if we get kind of devolved into a discussion, I'm going to hopefully kind of keep us on track as much as possible. But like I said, I just want to open it up first to questions from the room. Feel free to post your questions in chat, or if you want to unmic yourself and actually talk, that's totally fine too.
Could we go over activation energy and catalyst?
Sure. So activation energy and catalysts apply most to when we're talking about chemical reactions. And so catalysts, if we're talking about these reactions occurring in the body, just take into account that when we say the word catalyst, the types of proteins in the body that actually are catalysts are enzymes. So enzymes carry out this catalytic activity. So sorry, want to share my screen here with you. I just wanted to pull up the right place. Okay, so this is probably the best image. So I keep seeing wink, wink, star, stars here. I will be asking you about catalysts and enzymes and kind of how they're similar and what they do for reactions. There is a question there. So when two molecules are going to interact with one another, what the body has done is has created these proteins called enzymes. What enzymes do is they actually hold the two molecules that are interacting in a energetically favorable conformation. And so because of that energetically favorable conformation, it actually requires less energy for that reaction to take place. And so activation energy is the energy that's required for those two molecules to interact. So in a normal type of reaction, what would need to happen is you'd have to have those two molecules maybe serendipitously interact with one another. And so one way you could cause two molecules to serendipitously interact with one another is by increasing the heat, because you would increase the kinetic motion of the molecules. But for what we're talking about here, the ways to lower activation energy is by utilization of a catalyst or enzyme. That's going to be the way that you can do that. So it holds it into that conformationally kind of favorable way, and those two reactants are able to react with one another, and then they create their products. Activation energy just again lowers the amount of energy that's required for a reaction to take place. Enzymes are just organic catalysts. Yeah, I think that'd be probably fair to say. Catalysts can be a variety of things. So catalysts would be kind of like the bigger category, and enzymes would be kind of the biological type of catalyst, because you can have inorganic catalysts as well that you add to reactions. So anybody who's taken a chemistry class, you maybe have added a catalyst to reaction, and that's not a biological system. So you can add just reactants like that or a catalyst like that. But in a biological system, enzymes are the proteins that act as catalysts by holding those molecules in energetically favorable reactions. Does that answer your question, Tessa, or do you have kind of a more specific question?
Yeah, that answers it. Thank you so much.
Yeah. I would say because of the Type of material that we've covered in this first part, we haven't gone over a ton of anatomical structures. So it's going to be probably more towards the function side really than the anatomy side. So I'm not going to have you do a lot of labeling. Like chapter 5 is probably the first place we really start to delve into anatomy where we're looking at different levels of the dermis or the epidermis. Again, it's a little bit more anatomical, but for them, I would say focus more on like the functional side of things, just because when we're talking about things like atoms and cells, I guess cellular anatomy is kind of important, but I'm far more interested in what the function of what those molecules or those organelles are doing rather than the anatomical structure of them. That will probably shift in future chapters, however. So once we get into like our skeletal, muscular types of chapters, you'll probably see a lot more anatomical types of questions. It depends on what the focus of that chapter is. These chapters are kind of spaced out, so they kind of give focus to both of those. So we'll try to evenly represent those on future exams, but for this exam, probably more function. Uh, well, honestly, it's going to depend on how today goes. If today seems fruitful, useful for the students, if I get positive feedback from you all, I'm more than happy to do review sessions. I think it can be really helpful and help mitigate people's anxiety about the exam because I feel like they're getting a clear understanding of what's on the exam, and I'm more than happy to do that. So, yeah. Yeah, like I said, if you find it useful, then yes, I'd be more than happy to schedule review sessions for future exams. Okay, other topic related questions?
Can we go over the anatomical planes and the sections as well as the directional terms?
Sure. So this is kind of going all the way back to lecture 1. So this is, I mean, pretty much just memorization for the most part for the planes and structures. Oh, I'm just going to click this guy up here. You guys can see my screen, right? Wink, wink, nudge, nudge. Check this out. There's some information here that might be useful. Okay. For the systems of the body, I've not written any exam questions about the overall systems. So you're not necessarily going to see a question about a system that we haven't covered in class, just because I feel like since we're going to be going through those other systems in later chapters, it's kind of not really the overall focus of what we're talking about right now. Maybe spend a little bit of time, sorry, you asked about anatomical planes, understanding positive and negative feedback and homeostasis. Okay, so types of anatomical terminology is really what you were asking about. So body positions, regional names, directional terms, planes and sections, and body cavities. So these are ultimately just different ways in which you would describe the body to maybe an anatomist, or if you're a physician, to other physicians. And so anatomical position is essentially is the standard for how we relate the different regions to one another. So if somebody says to you anatomical position, they're talking about somebody facing forward to you, the upper extremities placed at the sides, palms are turned forward, and so any of the other kind of I guess terminology you would use for relations of body parts to one another, this is the anatomical, like the baseline that you're kind of measuring or giving those placements against. So in terms of like the regional names, I'm not going to go through all of these smaller names. Kind of these bigger ones are probably the most important that you see. So cephalic, cervical, upper limb, lower limb. We're not going to go into these smaller non-bolded regions per se. Directional terms. Now, this is important because this is where you're giving a direction based on relationship from one body part to another. So in this case, the fingertips would be distal to, let's say, the humerus. And so you need to kind of know the different relationships and directionalities between the two regions that you're talking about. So distal and proximal are often used when you're talking about the appendages, when you're talking about kind of top to bottom of the entire body, or you're talking about superior and inferior. If you're giving a relationship of an organ or a structure using the midline as your basis, you would use the terminology medial or lateral. So it just kind of depends on the two structures that you're relating to one another and the terminology that you would use based on that. Does anybody have any further questions about that? Because I feel like that maybe could be confusing.
I kind of do. Sorry. So you emailed me this, but I'm a little confused. What's the difference between like the directional terms and then like the supine, dorsal, and ventral position?
That would be more, I guess, relationships, like how a body was positioned if you were looking at it in placement. Directional terms are, I guess, are more a association of different regions, and then the supine, dorsal, ventral, you're talking about kind of the, if you weren't looking at somebody in anatomical position, that's how you would describe your vantage of them. Does that make sense? So if you were looking at somebody's back, you'd be looking at their dorsal side versus if you're using directional terms, you would be looking at somebody in the anatomical plane.
Okay, wonderful. Thank you so much.
Yeah. Planes and sections are another maybe wink, wink, nudge, nudge. So planes and sections, if you're going to actually, let's say, do histology on an individual, if you're doing the frontal plane or coronal, it'd basically be slicing something, if somebody was in anatomical positions, from their nose to the back of their head. So those would be frontal or coronal sections that are being taken. The midline, mid-sagittal plane is through the midline. So if you're going to cut somebody down kind of the center of their body, that would be the mid-sagittal. If you were going to offset it to either side, so let's say if you were going to cut here or here, those would be considered the parasagittal planes. The transverse plane is if you were going to essentially cut somebody from inferior to superior. And an oblique plane would be kind of an offset plane, not necessarily in any of the 90-degree planes that we're talking about. So this is this planes and sections are kind of, this is a nice example. I just like it because it's a brain, but this gives you kind of a good understanding of how those different sections would look if you were slicing a brain. A mid-sagittal, you'd be cutting your corpus callosum. A frontal or coronal slice, you'd be slicing the brain from the olfactory bulb to the I should probably stop saying those words. From front to back, and then your transverse again from superior to inferior. So just to kind of answer some of the questions that are popping up in the chat. So the person who asked about planes and sections, do you feel like that was a pretty decent overview? Because I don't want to move forward unless you feel like that question was answered completely.
Yeah, that's good. Thank you.
Okay. So what will the exam focus on most? I mean, that's kind of a difficult question to answer because right now the way that I've written it, since we have 5 different chapters, we have 50 questions, I am essentially writing 10 questions for each chapter-ish. Some of the chapters have, if you go to the study guide for exam 1, I wonder if I can you see this?
We can't see a study guide.
You can't see a study guide? Okay.
You can only see the PowerPoint.
Yeah.
Okay. That's totally fine. So as I'm writing the exam.
Now we can see it.
Yeah, sorry, I had to just do a new share. I'm basically going back and forth to the study guide and looking at these questions and writing my questions from this. So some of the chapters have more points. So let's say chapter 2 has more points, then I'm probably writing a few more questions for this. And I'm not writing a question for every single point, but I am trying to, again, not lead you astray saying, this is important and then not testing you on it. I really am trying to stick as close to the study guide as possible, even as I'm writing the exam. So I guess that's kind of the best way I can describe what the focus will be is I'm pulling it from these study guides. And so if it's focused here, that's the type of question I'll be asking. There will be labeling of, there's one question that I've written so far that is a labeling of a diagram question. And I think I just showed you that diagram not too long ago. It's in that chapter 1 slide. So yeah, just gave that one away. So, but in terms of other structures, no. Again, kind of as the question was earlier, if this is going to be more structure or function, I think this is far more function than structure. In future exams, when we're looking at the skeletal system and the muscular system, then you're probably going to see more labeling. The only other place I could foresee a labeling question being is if we're looking at the different layers of the integumentary system. So make sure you feel comfortable with the layers of the integumentary system. And not the smaller layers, like just make sure you know the epidermis, the dermis, the hypodermis, things like that. Parts of mitosis, just I would say like a 1-sentence description of each step of mitosis. And what happens. It's not necessarily a labeling question, but make sure you can recognize what the cell looks like in those stages of mitosis. So I'm not going to necessarily pull the mitosis slides up, but again, prophase, metaphase, anaphase, telophase. Make sure you feel comfortable with the ordering of those phases, kind of, again, a 1-sentence description of what happens in those phases. And then definitely in your notes, maybe you just make a drawing of what the cell looks like, where the chromosomes are distributed during those phases. Different types of junctions. Okay, that would be chapter 4, I think.
On some of these, like relate the anatomical names and the corresponding common names for various regions of the human body. Are you going to do like, you'll have a list of the anatomical names, and then we have to do a drop-down menu to pick the matching?
I didn't write a question like that yet. No, you won't need no, you won't need to do that.
That's just the one I saw right off the bat, and I thought that one would be a good one to ask. So it's just going to basically be, here's our question, A, B, C, or D type thing?
Yep, A, B, C, or D. And then I've written a couple, all of the above, none of the above types of questions. So just make sure you feel like you can easily distinguish between things that might like look the same. I know it's probably not something students like to hear, but I will make words that look very similar to as an option, so to make sure you feel comfortable with the names of things and the specific names. Yeah. Oh, another thing maybe worth wink, wink, nudge, nudge. Remember when we talked about protein synthesis and the different types of steps in protein synthesis? Addie, I'm coming back to your question, I promise. Definitely know those, the steps to protein synthesis, and then the macromolecule intermediates. Okay, so the different types of cell junctions, just going to Addie's question. So cell junctions are essentially the way when you've got an entire tissue, the cells kind of how they are glued together. So there are different ways that cells are glued together depending on what the types of cells that are interacting with one another are. So the first type of cell junction that we see here is a tight junction. Tight junctions are the types of junctions that basically glue two epithelial cells together. So you have your epithelial cells that create a barrier between your intestine and the rest of your body. So you want to make sure all those nutrients aren't just leaking through to the underlying basement membrane or the connective tissue. So in order to make sure that doesn't happen, your cells have to create these tight junctions with one another. So as you can see in this figure here, there are these transmembrane proteins that kind of weave together these tight junctions. And again, these tight junctions are so tight that ions or fluid or what have you cannot necessarily diffuse across that space between those cells. So they're holding those epithelial cells nice and tight together. So that's one type of junction. Adherence junctions are another type of junction. You can see here that it's a little bit more structured. You just don't have those transmembrane proteins that are holding together the cells. But similarly, they can be on adjacent epithelial tissues. And again, you've got multiple cells that are kind of creating a network And so here you have this microfilament actin, which we talked about when we were talking about the different types of structural proteins for the cell. And then those cells get kind of glued together with these transmembrane proteins, in this case, a glycoprotein called cadherin. And if we go back to chapter 3, when we're talking about the different types of molecules, glycoprotein is exactly what it sounds like. It's a protein with a sugar attached to it. And so that sugar helps kind of the interaction of those two cells, and they're anchored to proteins, and those proteins are embedded in the membrane. So it just kind of creates a nice interlocking attachment of those cells. And so here instructions, you can see here have very specific points of contact between the two cells at that adhesion belt. Desmosomes, as we look at here, are different in the fact that they are anchored not to just that belt, but they have these little, I guess, almost like spaghetti-looking things here are intermediate filaments of keratin. They just don't terminate here; they actually extend through the entire cell. So they're kind of a dense meshwork that kind of holds the entire cell structure together. So if you have cells that have desmosomes that are connected to one another because they're kind of anchored to those meshes that are keeping the structure of the entire cell, if you have cellular movement, those desmosomes are kind of aiding and anchoring those cells to move together in that cellular movement. Similar to what we just saw with the adherens junctions, you have a protein that is based on the cell, those plaques, as well as the transmembrane glycoprotein. This glycoprotein is slightly different. The protein, again, is going to be the transmembrane part of the structure. The sugar is going to be attached on top. So the sugar is kind of like the interlaced part that you see here that allows those two desmosomes to actually interact with one another. This is an example of the hemidesmosome. So it's like half of a desmosome. So you still have those keratin intermediate filaments that are anchored, but this is the type of junction that you would see between an epithelial cell and the basement membrane. So this would be on the basal surface of the epithelial cell. So in an epithelial cell, you have a basal side. That's kind of the basement, the part, right? The bottom of the cell. The apical side's up at the, hanging out in the fluid. And absorbing things. And so here you can see you've got these transmembrane proteins, or these glycoproteins again, that are anchoring that desmosome to that basement membrane. And lastly, you've got gap junctions. Gap junctions are a different type of junction. These are used more in signaling. The advantage of gap junctions is that they create these pores, and these pores allow for movement of ions, whether they be positively or negatively charged, to flow through those channels. And so it's one way in which the cell can communicate very quickly with its neighboring cell. Gap junctions create electrical synapses. We will talk about that more when we get to the central nervous system. It doesn't. Gap junctions exist somewhat in the central nervous system. More so, we see chemical synapses, but gap junctions again allow two cells that are neighboring to one another the ability to communicate via movement of ions. The proteins that allow the movement of ions across the gap junction are called connexins. So those are the proteins that are embedded in both of those cells. Those are transmembrane proteins that allow the passage of ions. Transmembrane proteins that allow the passage of ions that are integral proteins. Okay. So did that answer the question about the gap junctions? I think that was Addie that asked that.
Could you go over the different types of cells?
Yes. Samantha, would you in the chat write the specific types of cells? Whether are you talking about all the cells in the body or a specific type of subtissue cell? Write that in the chat because it's rapidly becoming my to-do list. Okay, so we need to go to chapter 3, I believe. Okay. So ATP. ATP stands for adenosine triphosphate. If we kind of go back to, I don't even know if I need a figure for this. So, okay. So adenosine triphosphate. And adenosine triphosphate essentially means that there are 3 phosphate groups that are attached to a molecule of adenosine. Now, the reason ATP is important is because in each one of those phosphate groups, there's actually a ton of chemical energy in those bonds. So when a bond is broken for ATP, energy is released. And so this is, you can think of ATP as being the energy currency for the cell, and each one of those phosphate groups that gets added kind of increases the value of ATP. So if it has 3 phosphates that are attached to that adenosine, that means you have a very high value energy molecule that's able to be used in reactions. Now, once that molecule gets used in a reaction, it loses that phosphate group, and instead of being adenosine triphosphate, you get adenosine diphosphate, ADP. And so at that point, you kind of have like a mid-range energy level molecule. It can still lose another phosphate group, but it can also gain a phosphate group to make it high level energy again. And so the cell has processes to do that, to add that phosphate group back on. And then if you lose both 2 phosphate groups, you get adenosine monophosphate, again, which sometimes happens in the cell, but the cell makes sure that it replenishes those phosphates and adds them to adenosine again. What we know is that mitochondria is the powerhouse of the cell, and because ATP is the energy currency of the cell, ATP is generated in mitochondria. We are not going to go through the process of how mitochondria makes ATP. That's a whole nother level of chemistry I'm sure your brains do not want to handle right now, but just recognize that ATP is made in mitochondria, and the energy of ATP is stored in those phosphate groups. Dawn, does that answer your question? Okay. So Audrey asked, well, we have to know the organelles in the cell and their functions. I would say know a 1 maybe sentence description. So I would say spend some time maybe just drawing a cell, draw the organelle, and then off of that organelle, write its function. I think that can be really helpful for a few reasons. One, it kind of gives you a sense of where things are organized in the cell, and then also what the function is of that particular organelle. Kind of keep going back to this idea of the cell being a protein factory, that the blueprint's kind of the head idea man in the cell is a nucleus, and that contains DNA. DNA is going to be transcribed into RNA in the nucleus. Then RNA is going to get shuttled out of the nucleus into the cytoplasm, and then it's going to go to ribosomes, either on the endoplasmic reticulum or the smooth or free ribosomes. And the ERs, or the ribosomes, are basically going to be the production mechanisms for making the protein. And then once the protein is made, it basically gets shipped via the Golgi apparatus, and the Golgi apparatus tells the cell where to send those proteins that have been made. And then that's the last step of the process, is those proteins end up different places in the cell carrying out their different functions. So if the protein factory analogy helps you, you actually cover a lot of the different organelles in the cell. I mean, you could think of the mitochondria, again, because of the energy producing organelle in the body. You could think of it as like the cafeteria that feeds all the workers all of the energy that they need to do, or the lunchroom, maybe, I guess. So yeah, I think that analogy always works for me. Hopefully it works for you, and it kind of gives you, I think, a good sense of what the function of the cell is. But yeah, maybe you probably just write a 1-word description of all the organelles, so you know what they're doing in the cell. Types of connective tissue. Okay, types of connective tissue. That would be chapter 4, part 2. Okay. So types of connective tissue, going over here right now. Okay, so connective tissue is interesting because it exists in kind of both a cellular state and an extracellular matrix state, meaning that it's not necessarily like universally very similar in some ways. Connective tissue, you know, connects a lot of different parts of the body. So when we look at connective tissue, this really is probably a great overview slide to kind of study from, because it gives you a one-sentence description of what each of one of these types of tissues are, and it also kind of gives you a visual as to what they look like. So I revisited, oh, screen sharing is paused. Why are you paused screen sharing? Oh, there we go. I'm going to try to reshare this. It wouldn't be, as some might say, it wouldn't be human form and function if there wasn't a technical difficulty, right? Okay, so the different cell types that we see here for connective tissue. Like I said before, I think often when you're learning things like this, characterizing things that are similar and different makes it easier. So I'm gonna again do the same thing I've done before, is kind of circle the different cells, because all of these cells fall into the category of being cellular. So cells are round, so I like to circle them. So here we have all of the different cells. So connective tissue has macrophages, fibroblasts, mast cells, plasma cells, neutrophils, eosinophils, and adipocytes. So we'll use orange. Orange is going to represent the immune cells here. So macrophages are a type of immune cell that eat things via phagocytosis. Wink, wink, nudge, nudge. Mast cells are another type of immune cell as well. They're very similar. They also weren't listening. They also function during inflammation and kill bacteria. You'll see that plasma cells secrete antibodies and neutralize foreign bodies. Neutrophils are also immune cells and destroy microbes by phagocytosis, similar to the way that macrophages do. Eosinophils are also white blood cells and attack parasites and also mediate allergic responses. So those are essentially the different types of cells that are considered connective tissue. So again, our big characterization, right? Fibers, cells. And then if you went down to like another level of characterizations, you would be like immune cells and then cells that do something else. So the cells that do something else would be things like fibroblasts that secrete ground substance, which is actually another type of connective tissue. I'll use that term very loosely there because it doesn't necessarily have a kind of tissue type of characteristics. Adipocytes are another type of connective tissue, and those are essentially fat cells. So they insulate your body. They can be used for energy resources. So yeah, so then we've covered all of the types of cells, and then other types of connective tissue. The reticular fibers are made of collagen. Again, they're forming more of a structural form of connective tissue. Collagen are another type of fiber produced of collagen, again, giving us some elasticity to our body. Interesting, we also have elastic fibers made of proteins elastin and found in skin, blood vessels, and lung tissue. And kind of like the last thing that I'll circle here in a completely different color is going to be ground substance, because this is essentially the material between the cells and fibers. Think of it almost like the cytoplasm of the body kind of things. It carries out the same functions like that the cytoplasm would in the cell. So it has a bunch of organic molecules that hang out in it that are important for some of these other cells to survive and thrive, and binds them together. So it's a substance that again essentially exists between blood and cells, and it's produced by the fibroblasts. So yeah, the thing that I circled before, and I was like macrophages, wink, wink, nudge, nudge, eat things by phagocytosis, which is a type of active transport. Okay, types of connective tissue. So Aiden, did that answer your question? Okay, cool. Cells. I guess if we go like the biggest types of cells in the body, it's hard because I want to characterize the types of cells as the same thing as types of tissue, but it's not necessarily the same thing because you have tissue types, and then those tissue types are kind of further distributed into types of cells. So I almost want to say the types of tissue that you see there. I guess we do have a slide that talks about the diversity of cell shape, which let me see if I can find. I gotta erase all that writing I did. I'll see if I can find that slide because that might be what you're thinking of. Erase, erase, erase. Goblet cells, like the shape things that I don't know the names for. Goblet cells. Maybe let's go to chapter 4, part 1. So types of cells, since the chapter 4 is primarily focused on tissues, the 4 types of tissues, right? You have epithelial tissue, you have connective tissue, you have muscular tissue, and you have neurons. Now, those last 2 that I mentioned, muscular and nervous system tissue types, you can basically not spend your time focused on those because we're going to go over those in later chapters, so there's really no point in asking you specific questions about that now. We just went over all the different types of connective cell tissues, and so really maybe the only other place where we're seeing cell types is in epithelial tissue. So maybe that's another great place to go. And I should have opened up that other review. Let's see. It was like the first lecture or 2. Okay, that's good information to have. Lecture 1, lecture 2. Okay. Okay, go back to epithelial tissue is what I'm taking it as. This. Okay, can you see classification of epithelial tissue? Okay. So I probably shouldn't. So in terms of epithelial tissue, if you want to get down into like the nitty-gritty of each one of those individual cell types, probably we only talk about the specific functions. Like you mentioned something like goblet cells. If we talk about epithelial cells, it's just important to recognize that there's a directionality to them and that they have a basal surface and an apical surface, which we've talked about multiple times. This is all part of the epithelium. Then you have your basement membrane and your connective tissue underlying. This is kind of a nice synopsis slide of the different types of tissues that we talked about in chapter 4. In terms of like the shapes are useful because each one of the different cell types that epithelial tissue have are described. If they're simple, pseudostratified or stratified, that's the arrangement of the way the cells are put together. So goblet cells would, for example, would be an example of a pseudostratified cell in terms of the arrangement of the layers, and they would be columnar. You can see that you can also describe them via cell shape. So you have squamous, cuboidal, columnar. So those are the 2 ways in which you describe an epithelial cell. If you are interested in examples of all of those different arrangement shape combinations, this table 4.1 in your textbook is a great resource. I actually took all those tables out of 4.1 and put them into the study guide review PowerPoint that's posted onto CourseSite. So if you want the PowerPoint version of what those tables look like, you can go to chapter 4, part 1, study guide review, and all of those tables are represented there. I'm not going to ask you specific examples of those. I think that's kind of getting into the details. And honestly, I think if I told you that you had to do that now, you probably would freak out and rip your hair out because there's just a lot of different combinations there, and understanding an example of each, I think, is a tall order. But when we talk about, you mentioned goblet cells, that's just an example of glandular epithelium. So we talked more specifically about those because of their function, because we were talking about types of secretions. So they are going to secrete they're going to be exocrine types of glandular epithelium. And so endocrine and exocrine glands are worth just maybe looking over, and the types of secretions that are secreted from those glands. Endocrine glands, again, are kind of sending out chemical messengers that are traveling long distances from the body. So that's why hormones are a great example of endocrine glands. Exocrine glands are secreting things in a far more local sense. And so sweat, oil, and earwax, these are all things that we talked about in chapter 5, are going to be local types of secretions that act in that one region. Goblet cells is just an example of an exocrine gland. So glandular epithelium, there you go, you've got your goblet cell, sweat glands, oil glands, and I guess salivary glands. And these are essentially the types of exocrine ducts that you can see for the different type of secretions. So skin, these are the different types of glandular epithelium. I feel like I'm kind of getting into the weeds here and giving you a lecture again. So I'm going to pause for a second and see if somebody has a more focused question, because I don't want to sit here and re-give a lecture if no one has this actual question. You're not going to fail. I think you'll be surprised. I think if you don't get overwhelmed with the details, which I feel like you might feel like there are right now, like if you go over that study guide and you feel pretty confident with those questions, if those questions don't overwhelm you, then you'll be absolutely fine. Again, hopefully those questions are giving you kind of some type of structure. And like I kind of described in class yesterday, like just making a single page, and this is how I study, and it's not for everybody, but just take a single kind of blank white piece of paper and kind of just start putting together the structure of the entire chapter. Like separate things out. Like for example, chapter 4 is a great example. Take that piece of paper, split it into half, make half of it connective, make half of it epithelial, and that's the kind of the overall larger structure for chapter 4. Now, not all chapters are that easily divisible, but again, if you know that big picture, then you can start to fill in the different details, right? So if you have, this is connective, this is epithelial tissue, you can be like, these are the different types of cells in connective tissue and epithelial tissue. These are the diseases associated with connective tissue or epithelial tissue. Yeah. Other questions or concerns? We have about, I think, 10 more minutes here, 15 more minutes. I just want to make sure I'm using this time in the most focused, useful way for everyone. The difference between pseudostratified and columnar cells would be essentially the amount of basal and apical exposure there are. So for columnar cells, there would be equal amount of exposure to both the apical and basal sides. But you'll also recognize here that you've got, this is an arrangement of layers and this is a cell shape. So you can see that those two things can almost be combined. Pseudostratified, you see, that are not necessarily equally distributed in the basal and apical side. Some of them have far more area distribution to the basal side versus the apical side. So this little guy down here would be an example of a cell that has a far more higher area coverage to the basal side. And if we look at maybe this guy, or this guy would be a great example, you can see far more apical exposure than basal. So pseudostratified isn't necessarily that straight up and down columnar shape. But you also see that when we look at the naming combinations, pseudostratified can essentially only be columnar because you're looking at both the arrangement and the shape. So pseudostratified have to be columnar. But columnar can also be the different simple pseudostratified or stratified. So columnar can actually be a lot of different arrangements, but in terms of pseudostratified, it has to be columnar in shape. Good question. Okay. Other questions or concerns? I feel like I've given you a lot, kind of hints here. So, yeah. I mean, I would have to say there's probably at least like 8 to 10 questions that you should know of a focused question that's being asked. No problem, Carson. Have a good day. But yeah, like I said, as you're looking over the material, does anybody? That's 20% given. You know, there's got to be kind of a level of I can't just show you the exam right now. That's just you know, not the way this works. Oh, remember yesterday when we talked about mucosal membranes? That was a wink, wink, star, star. Okay. As you're going through your notes. Okay, I'm trying to think. Kind of giving you the tips as to how I would study. Far more function than structure. Again, some of all of the above, none of the above, multiple choice. Some of those questions are sentences where it's almost like you're filling in the blank. So there'll just be an option for you to fill in the blank using a selection for one of the words below. Trying to think of what else I can tell you about the exam. I'm trying to make it equal distribution across the chapters, but like I said, some chapters are a little bit lengthier, so there might be a few more questions dedicated to that. Yeah. Okay. So I'm gonna give you guys a few more minutes to see if anybody has any content questions. And if not, then we're gonna end up, I'm gonna end up letting you go. But, so if you have a content question, this is kind of your chance to pose it in the, go over protein synthesis. Happily, I will happily go over protein synthesis. Okay. Okay, sorry. Chapter 3. Okay, so in terms of protein synthesis, if you watched the lecture, I mentioned that protein synthesis is essentially called the central dogma. The central dogma is kind of the cornerstone to biology because it talks about the cell, or cell biology at least, because it talks about the process of how you get from DNA to RNA to protein. So those are kind of the 3 macromolecules. So you will see a question dedicated to this. I told you in the lecture, and I'm telling you again right now. You will see a question on the central dogma on this exam. I'm just looking for the, perfect. Goodness. Okay. So this is essentially what I'm talking about when I say the central dogma. So you have in the nucleus, you have DNA, right? And the first step of that DNA is to be transcribed into RNA. So just make sure when we're talking about the processes, you feel comfortable with the terminology transcription. And then the next step in the process is once this RNA has left the cell, it gets exported out of the cell and comes and hangs out with this ribosome. And that ribosome essentially is going to help that RNA be translated into protein. And so that's ultimately kind of the central dogma in a nutshell. The only kind of thing we're leaving out here is the fact that DNA can make more DNA via replication, but I'm not going to ask you about that because it's outside of the scope of this course. But these are the steps of protein synthesis and the processes. So DNA becomes RNA via transcription. RNA becomes protein through the process of translation. I know this looks terrible. It's actually written out here on the slide, so you don't have to try to read my chicken scratch. You've got transcription and translation are the two processes. Okay. I would say probably, because I'm using the study guide to write the exam, the study guide is probably more useful in a focused way. If you feel comfortable with the study guide and you want to just test your own knowledge, then maybe the Wiley Plus can be useful for testing your knowledge. So I would say probably spend a majority of your time going over those study guides, and then once you feel comfortable with that, you're like, okay, I've got this, I'm going to do great. Maybe go in and do some more Wiley Pluses, because I think that could be a useful just test of your knowledge. Okay, go over the macromolecule intermediates. And so the 4 macromolecules that we have, or that the cell is composed of, are going to be, hey, maybe I'll ask you a question. Can somebody throw out a macromolecule? Macromolecule.
Is it like lipid?
Yeah, so lipids would be an example of a macromolecule.
Protein?
There are 4 macromolecules. Proteins are a macromolecule, yes.
And
So lipids.
carbohydrates?
Carbohydrates are a macromolecule, so lipids, carbohydrates, proteins, and we're missing one.
I don't remember.
Okay, what else is in the cell? DNA. Okay, yeah, so totally DNA. DNA kind of falls under the category of a nucleic acid. Because you've got both DNA and RNA, those are both types of nucleic acids. So you have nucleic acids, proteins, carbohydrates, and lipids. And so those are macromolecules, or also called polymers, meaning that they're put together of smaller subunits. And so each one of those macromolecules has smaller subunits that make them up. So for proteins, what are proteins composed of? What are the smaller units of proteins? Amino, like the question mark. No, say it with like, you know, gusto. I know the answer. Amino acids, yes, are the smaller components of proteins. So amino acids are joined together via peptide bonds to form proteins. So those are the small subunits that form proteins. So we've covered proteins. Lipids. Lipids are not as necessarily, I guess, clear in terms of what the smaller structure of a lipid is. A lipid can be composed, basically has some type of backbone, maybe a phosphate backbone or a glycerol backbone, and then it has these long chains or tails off of those, and those long chains or tails are what make up that lipid. What's interesting about lipids is that they can be monosaturated or sorry, not monosaturated, they can be unsaturated or saturated. So that's a wink, wink, nudge, nudge. Know what a unsaturated versus a saturated lipid, what that is. Okay, so we've got difference between here we go. This is great. Okay, can you see the fatty acid in front of you? Okay.
Yes.
Okay, so fatty acids can be saturated or unsaturated. So saturated fatty acids this is the way I keep it straight. Saturated fatty acids are saturated with hydrogens, meaning that every carbon has a hydrogen bonded to it. There are no double bonds. It's saturated with hydrogen, no double bonds, saturated, saturated fatty acid. And unsaturated fatty acid is not saturated with hydrogen bonds. So here you can see that these two carbons actually aren't saturated with hydrogens. I should just use my little drawing things. So there are two hydrogens that possibly could be bonded here, but because there's no hydrogens, you form a double bond. So unsaturated fatty acids form a little kink in their leg. So because of that, saturated fats are solid. Unsaturated fats are liquid in their phases. So that's one kind of structural difference of the two. So difference between saturated and unsaturated fats. There. Like I said, you can, fatty acids don't necessarily have like the smaller subunits that we see with some other macromolecules, but they generally have some type of backbone. So in this case, you see a glycerol backbone and then a type of fatty acid molecule. In this case, it's palmitic acid, but I would not ask you to get that specific in terms of understanding the different fats, because again, saturated, unsaturated, wink, wink, star, star. This stuff, not so much. Okay, different other macromolecules. So we've gone through proteins, we've gone through lipids. Carbohydrates are made up of essentially sugars. What would you call the smallest subunit of a sugar? Because sugars can be linked in long chains. So what would you call that? Monosaccharide, exactly, yes. So a single ring structure of a sugar is a monosaccharide. So if you join together two monosaccharides, you would have a what? These are monosaccharides. Polysaccharides? Yeah, I mean, if you join together a lot of monosaccharides, you would have a polysaccharide. But if you just
Disaccharide?
Yep, exactly. So if you join together two monosaccharides, you have a disaccharide. So disaccharide, wink, wink, nudge, nudge on the disaccharide note that we just made. So you can see examples here. I think sucrose might be the disaccharide that we talk about. Anyway, yeah, sucrose. Okay, so disaccharides, monosaccharides are the components of carbohydrates. Okay, so the last one we have to talk about is the nucleic acids. What's kind of an individual nucleic acid composed of? Nucleotides, yes, exactly. So nucleotides are kind of the components of nucleic acids. If you look at an individual nucleotide, it is composed of a phosphate group, a pentose sugar, and a nitrogenous base. Those pentose sugars can be either deoxyribose or ribose, depending on if it is DNA or RNA. Wink, wink, nudge, nudge, the difference between these two. You don't need to know the structural difference, but just recognize which type of sugar goes with which type of nucleotide. So yes, for nucleic which type of sugar or what deoxyribose goes with DNA, ribose goes with RNA. No, that. So the sugar phosphate backbone you can see here. Man, this is recorded. Everyone is gonna go back and just like rewatch all the wink, wink, star, stars, be like, oh, this is exactly what she said. But you know, hopefully, again, that helps decrease a little bit of test anxiety. So for nucleic acids, you have there will be an uproar. There's a phosphate group and a sugar group, and it's just repeated patterns of those sugars and phosphate groups that form the backbone. That's when we say a sugar phosphate backbone for the nucleotides. And then each one of them has a novel nitrogenous base that gets attached, and those nitrogenous bases are composed of CTU, A or G, and that's kind of how the genetic code is essentially created. You've got that backbone that's very similar, you know, phosphate, sugar, and then the nucleotides kind of hanging off of it are what make the sequences of DNA unique. Okay, we've got essentially 1 minute left, so if anyone has any questions, now's a good time to ask. for those of you who've hung out, I assume that you have, Samantha, you're just, yeah. Yeah, I imagine, no problem. Yeah, I'll just send it to you right now. No. So for those of you who've hung out, I assume you've found this review useful. Will there be any extra credit questions on the test? Probably. I usually like to throw one on there, maybe two. So yes, there probably will be extra credit questions. Maybe content related, maybe just randomly related. We'll see. Other questions or concerns about the exam tomorrow, about the content? Random would be nice. I know, but I feel like random questions about kind of the world is like you're selecting out for people's content knowledge or awareness. So it's just like, like what's going on in Missoula today? Is it not so smoky, very smoky, a little smoky? I mean, that would be one that everybody could probably answer correctly. I guess maybe. Okay. Anyway, if you have questions between now and tomorrow, just go ahead. Feel free to send me an email. I'm going to be up probably pretty late tonight trying to update grades. So I will be checking my email regularly. Okay. Does anybody have any other questions or concerns before I let you go?
Thank you so much.
Yeah. Thank you guys for coming. I mean, I appreciate your time because if no one came, I wouldn't do this again. So you basically have bought future review sessions for the class. So your classmates should thank you. Yeah. Awesome. Okay, great. Good luck on the exam tomorrow. Yeah. And have a good day. Stay safe and stay healthy. And I'll see you soon.
You too. Thanks so much.
Thank you.